Lecture Notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 29. 14. Homeostasis. Cells function more efficiently if they are kept in near-optimum conditions. Cells in multicellular animals are surrounded by tissue fluid. The composition of the tissue fluid is kept constant by exchanges with the blood. In mammals, core temperature, blood glucose concentration and blood water potential are maintained within narrow limits to ensure the efficient operation of cells. Homeostasis has become the central unifying concept of physiology and is defined as a self-regulating process by which an organism can maintain internal stability while adjusting to changing external conditions. 14.1 Homeostasis in mammals. The word homeostasis means steady state. Homeostatic process keeps conditions in the body within narrow limits. Body cells only function normally within a narrow range of conditions. Their enzymes are sensitive to temperature and pH changes. They need materials for growth and respiration, the removal of waste products, and a specific composition of surrounding tissue fluid. Therefore, homeostasis provides body cells with relatively constant optimal conditions, independent of variable external conditions. 14.1-1 Importance of homeostasis. Homeostasis maintains optimal conditions for enzyme action throughout the body, as well as all cell functions. It is the maintenance of a constant internal environment despite changes in internal and external conditions. In the human body, these include the control of blood glucose concentration. 14.1-2 Principles of homeostasis. Homeostasis is the maintenance of approximately constant internal body environment, e.g., body temperature. The control systems must have a sensor monitoring the environment, which relays information to a control center that stimulates an effector to change the environment. The system works through negative feedback loops, since the action of the effector is monitored by the sensor and relayed to the control center, which then modifies the action of the effector. Homeostasis works through negative feedback loops. The action effector is monitored by sensors and relayed to a modifying control center. Homeostasis is usually maintained by negative feedback mechanisms. The body detects a change in a particular internal factor, such as core body temperature, and then activates a corrective mechanism to reinstate the normal level. Homeostatic control systems use negative feedback in which any change in a factor stimulates actions by effectors to restore the factor to its set point. 14.1-3 Deamination. Surplus amino acids cannot be stored, and so is deamination in the liver. Amino groups removed enter the ornithine cycle to form urea, which is removed from the blood in the kidney. Lecture Notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 30. Excretion is the removal of the waste products of metabolism and substances in excess from the body. For example, in the liver, deamination is the breakdown of surplus nitrogen-containing compounds, e.g. amino acids, which cannot be stored. The amino group, NH2, is removed from the amino acid, forming ammonia, NH3, through the addition of a hydrogen atom. This highly toxic ammonia is entered into the ornithine cyclical series of reactions, reacting with CO2 to form less toxic urea, which is taken to the kidney in the blood. The deamination of amino acids in the liver produces ammonia, which is converted into urea, the main nitrogenous waste product. Urea is excreted in solution in water, as urine. 14.1-4 Structure and function of human kidney. The basic functional units of the kidney are coiled tubes called nephrons. They function both for nitrogenous excretion and osmoregulation. Body temperature of ectotherms, e.g. insects, varies with the environment, whereas the body temperature of mammals can be decreased by physiological mechanisms such as vasodilation and sweating. Blood temperature is monitored by the hypothalamus. If the temperature falls, the heat gain center stimulates heating mechanisms in the skin, muscles, and metabolism. Increased blood glucose concentrations are detected by beta cells in the islets of Langerhans in the pancreas, which releases the hormone insulin. The remainder of the amino acid is used in cellular respiration. Mammalian kidneys function as organs of nitrogenous excretion, filtering urea from the blood, and osmoregulation, filtering excess sodium and water. Each kidney contains 1 million coiled tubes, nephrons, where urine is formed. Toxic substances in glomerular filtrate, e.g. urea, need to be removed from the body. Selective reabsorption in the proximal convoluted tubule, PCT, reabsorbs those substances present needed by the body, including glucose, amino acids, and sodium ions. These diffuse from the tubule into the surrounding cells, and sodium ions. These diffuse from the tubule into the surrounding cells which are adapted for. Lecture notes for A-Level Biology by Mohammad Rashid Chatha. 030046104457. 31. Reabsorption with microvilli to increase surface area and many mitochondria to manufacture ATP. Active transport then pumps these molecules into intercellular spaces. This maintains a concentration gradient, so diffusion can continue out of the tubule. From the intercellular spaces, the molecules diffuse into the blood capillaries surrounding the PCT. The removal of these soluble substances results in an osmotic gradient between the filtrate and the surrounding cells. This allows reabsorption of 80 to 90% of the water from the tubule via osmosis back into the blood. The loop of Henle and the collecting duct form a system known as the countercurrent multiplier, which enables production of urine more concentrated than blood, hypertonic. Na+ and Cl ions are actively pumped into the intercellular space, producing a concentration gradient into the medulla. H2O cannot follow because the ascending limb is impermeable. H2O moves out of the permeable descending limb via osmosis, increasing the filtrate concentration. Filtrate at the base of the loop is very concentrated. When it enters the ascending limb, Na+ ions are pumped out. This makes the filtrate at the top of the limb dilute again, allowing more H2O diffusion in distal convoluted tubule. The filtrate drains into the collecting duct, which flows back through the concentrated medulla. Temperature control is vital for enzymes to work at their optimal rate. Ectotherms, e.g. reptiles and insects, are organisms whose body temperature varies with environmental temperature. Temperature regulation occurs mainly through behavioral mechanisms, e.g. marine iguanas bask in the sun to heat up and lie on shaded, damp rocks to cool down. Birds and mammals are endotherms. They are able to maintain their body temperature above that of the environment by behavioral and physiological mechanisms. Cooling mechanisms in endotherms. Behavioral physiological increased convectional heat loss to the atmosphere, flapping ears, e.g. elephants. Increased convectional heat loss to the atmosphere, relaxation of hair erector muscles, trapping less air around the body. Increased heat loss via evaporation from body, panting, e.g. dogs. Increased heat loss via evaporation from body, increased volume of sweat production onto skin. Increased conduction of surroundings, wallowing in mud, e.g. hippopotamus. Vasodilation increases flow of warm blood to the skin surface. Lecture notes for A-Level Biology by Mohammad Rashid Chatha. 030046104457. 32. 14.1 to 5 nephrons. The kidneys remove metabolic waste and control water and solute levels in the body. As a result of these functions, the kidney also plays a vital role in the control of blood volume and pressure. Each kidney is made from around 1 million nephrons, narrow tubules closely entwined with blood vessels. The first filtrate has the same composition as tissue fluid. As it passes along the nephron, the composition of filtrate is altered by various active transport mechanisms that reabsorb some substances while allowing others to pass into the urine. A large amount of filtrate is formed, but over 99% if it is reabsorbed in the first convoluted tubule, mainly by active transport mechanisms and osmosis. Usually, all glucose and amino acids are reabsorbed into the blood. The movement of solutes from the loop of Henle creates a region of high solute concentration in the medulla, through which the collecting ducts must pass. As filtrate, now urine, flows along the collecting ducts, water leave it by osmosis. The resulting urine is hypertonic, more concentrated than body fluids. The second convoluted tubule is involved in several homeostatic mechanisms including the regulation of salt, water and pH levels. 14.1 to 6 Formation of urine. The high pressure forces small molecules, i.e. H2O, glucose, amino acids, sodium chloride and urea from the blood through the pores in the capillary, across the basement membrane surrounding the capillary into the nephron, forming. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 33. Glomerular filtrate. This process is called ultrafiltration. Larger molecules, e.g. plasma proteins, cannot pass through the filter. Selective reabsorption in the proximal convoluted tubule, PCT, of useful substances, e.g. glucose, amino acids, sodium ions, involves diffusion, facilitated diffusion, and active transport. PCT cells have microvilli to increase surface area and mitochondria to manufacture ATP in respiration. At one end of the nephron, the renal capsule, the kidney filtrate is formed by ultrafiltration, pressure filtration, of the blood. The kidneys produce urine by ultrafiltration and selective reabsorption. 14.1 to 7 Structure of Bowman's capsule. The Bowman's capsules contain dense capillary networks, glomeruli. The blood pressure is high inside these capillaries because, 1. Blood in the renal artery arriving at the kidney is at high pressure. 2. The efferent arteriole removing blood is narrower than the afferent arteriole supplying blood. The glomerulus is a system of dense capillary networks found in the Bowman's capsule. High blood pressure is maintained by the efferent arteriole being narrower than the afferent arteriole, resulting in ultrafiltration. Blood is brought to the glomerulus in an afferent arteriole. High hydrostatic pressure in the glomerulus forces substances through the endothelium of the capillaries, the basement membrane and between the podocytes of Bowman's capsule. The basement membrane acts as a filter, allowing only small molecules through. The filtrate collects in the Bowman's capsule and then enters the proximal convoluted tubule, where most reabsorption occurs by diffusion and active transport. Substances are also reabsorbed in the distal convoluted tubule and collecting duct. Endothelial cells that line capillaries in the glomerulus have many perforations, and podocytes have many slit pores that aid the filtration of small molecules from blood plasma into Bowman's capsule. 14.1 to 8 Roles of hypothalamus and posterior pituitary gland. The hypothalamus continually samples the blood, detecting small changes in temperature. When the temperature falls, nerve impulses pass from its heat gain center, which stimulates heating mechanisms in the skin, muscles, and metabolism. Conversely, the heat loss center stimulates cooling mechanisms. The hormone ADH causes the walls of the collecting duct to become permeable to water, meaning water moves via osmosis out into the surrounding blood capillaries. When the water potential of the blood drops, e.g. when we dehydrate, it is detected by the hypothalamus, which stimulates ADH release from the pituitary. ADH makes the second convoluted tubule and the collecting duct more permeable to water, so water leaves the filtrate and enters the blood. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 34. In the loop of Henle, the ascending limb is impermeable to water and the descending limb is permeable. Urine concentration is controlled by ADH, which increases permeability of the walls of the collecting duct. Increased blood glucose concentration is detected by beta cells in the islets of Langerhans of the pancreas, which release insulin. The insulin increases the uptake of glucose into cells in the liver, muscles, and adipose tissue. Uptaken glucose is converted to glycogen, proteins, or fats and broken down to CO2 and H2O. Decreased blood glucose concentration is detected by alpha cells in the islets of Langerhans of the pancreas, which release glycogen into glucose, which releases glucagon. The glucagon activates enzymes that convert glycogen into glucose, which is then released into the blood. Other hormones, e.g. adrenaline, also increase blood glucose levels. Blood glucose levels are monitored by the islets of Langerhans in the pancreas. If levels get too high, beta cells in the islets secrete insulin. If blood glucose gets too low, alpha cells secrete glucagon. Insulin, a peptide hormone, increases the permeability of cell surface membranes to glucose. It also appears to activate intracellular enzyme systems that convert glucose to glycogen, fat and protein. Glucagon promotes the breakdown of glycogen, releasing more free glucose into the blood. The liver is a large organ that plays a major role in controlling the composition of the blood. Animals are divided into two groups according to their ability to thermoregulate. Endotherms, heart from within, maintain a stable core temperature by behavioral and physiological means. Ectotherms maintain a relatively steady body temperature using behavior only. Humans, like all mammals, ectotherms. We also say that mammals are warm-blooded, maintain a stable core body temperature despite changes in the external temperature. To maintain a stable body temperature, heat loss must equal heat gain. Unless the climate is very hot, mammals regulate body temperature by producing heat as a result of metabolic reactions, and by controlling how much of it is released back to the environment. When mammals are too hot, they maximize heat loss. When they are cold, mammals try to lose as little heat as possible. Heat is transferred in four ways: conduction, convection, evaporation, and radiation. Temperature receptors in human skin provide an early warning system for temperature change, allowing us to thermoregulate by our behavior, altering position, clothing, etc. This often prevents the need for a physiological response. The hypothalamus can detect the temperature of the blood in the core of the body. If the blood temperature drops, the hypothalamus initiates corrective responses. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 35. Such as vasoconstriction and shivering. If the blood temperature increases, we respond by vasodilation and sweating. These are physiological responses. Extremes of both heat and cold can cause death. Death as a result of cold occurs as a result of hypothermia. This is common in people who have fallen into freezing water. The loops of Henle act to produce high concentrations of sodium and chloride ions in the tissue fluid in the medulla. The medullary tissue has a very low water potential. Water is reabsorbed from fluid in the collecting ducts by osmosis if the body is dehydrated. The water content of the blood is controlled by changing the amount of water excreted in the urine by the kidneys. This is done by regulating the permeability of the epithelial cells of the collecting ducts to water, and hence the volume of water reabsorbed from the collecting hypothalamus. If the water potential decreases, the hormone ADH is released by the posterior pituitary gland. ADH stimulates the increase in the uptake of water from urine by collecting ducts. 14.1 to 9 Cell signaling. Mammals keep their internal environment relatively constant, so providing ideal conditions for cells to carry out their activities efficiently. The maintenance of relatively constant internal conditions is known as homeostasis. Homeostasis balance requires receptors that detect external changes in conditions, such as the temperature of the surroundings, and internal changes in physiological factors such as body temperature and pH, glucose concentration, and water potential of the blood. These changes in internal and external stimuli are detected by receptors, such as temperature receptors in the skin and asthma receptors in the hypothalamus. The nervous system and the endocrine system coordinate homeostatic mechanisms. Cells is controlled by the action of the protein hormones insulin and glucagon, which are secreted by the islets of Langerhans in the pancreas. Insulin stimulates cells in the liver, muscles, and adipose tissue to take up more glucose. In the liver and muscle, it stimulates the formation of glycogen for short-term storage. Adipose tissue responds by converting glucose to fat for long-term storage. The overall effect of insulin is to decrease the concentration of glucose in the blood. Glucagon stimulates cells in the liver to break down glycogen and release glucose into the blood. The effect of glucagon is to increase the concentration of glucose in the blood. Insulin and glucagon are proteins and so cannot cross the membranes of their target cells. Both hormones bind to specific receptors in the cell surface membranes of these cells. The binding activates a series of changes in the cells that amplify the signal so that the responses of the cells are rapid. The 14th of January 11 biosensors and test strips biosensors use the same principle but produce a small electric current instead of a color change, which provides a direct digital readout. Immobilized glucose oxidase is used on test strips to detect glucose. The enzyme changes glucose to gluconic acid and hydrogen peroxide, which causes a color change in another chemical on the stick. The range of colors on the strip indicates the concentration of glucose in the urine. Test strips are used to test urine for a variety of substances, including glucose. The presence of glucose may suggest that a person has diabetes. 14.2 Homeostasis in plants. Homeostasis is the ability to maintain a stable internal state despite external changes in the world. Plants absorb water from the soil and use it to maintain homeostasis. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 37. 14.2 to 1 Stomatal response to change in the environmental conditions by opening and closing, and the regulation of stomatal aperture balances the need for carbon dioxide uptake by diffusion with the need to minimize water loss by transpiration. Stomata control the movement of gases between the atmosphere and the intercellular airspaces inside a leaf. They allow the inward diffusion of carbon dioxide for use in photosynthesis and the outward diffusion of water vapor in transpiration. Each stoma consists of 2 guard cells either side of a pore. The cell walls of guard cells have hoops of cellulose fibers that restrict their increase in volume to the ends. When guard cells become turgid, the cells push on each other to open the pore. Guard cells are highly specialized cells that respond to changes in light intensity and carbon dioxide concentrations inside the leaf. 14.2 to 2 Guard cell structure and functions. In general, guard cells open during the day and close at night, although this rhythm persists in continuous light and in continuous dark. The cell surface membranes of guard cells contains proton pumps that activity transport hydrogen ions out of the cells. This stimulates the inward movement of potassium ions down their electrochemical gradient. The potassium ions decrease the water potential of the guard cells, so water enters by osmosis, the cells become turgid and open the stoma. To close the stoma, the proton pumps stop working and the potassium ions flow out of the cells. This raises the water potential inside the cells, so water passes out by osmosis. The guard cells become flaccid and this closes the stoma. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 38. 14.2 to 3 Opening and closing of stomata. The opening and closing of stomata depend on the turgor pressure, caused by the osmotic flow of water in the guard cells. When the guard cells are turgid, they expand, resulting in the opening of stomata. When the guard cells lose water, they become flaccid, leading to stomatal closure. 14.2 to 4 Role of abscisic acid in the closure of stomata during times of water stress, including the role of calcium ion as a second messenger. Abscisic acid, ABA, is a plant hormone that is synthesized by any cells in a plant that contain chloroplasts or amyloplasts, especially in stress conditions. The presence of large concentrations of ABA in leaves stimulates stomata to close, reducing the rate of transpiration and so conserving water inside the plant. Calcium ions act as second messengers in guard cells in response to stimulation by ABA. Keywords: abscisic acid, ABA, adenylyl cyclase, adrenal glands, adrenaline, afferent arteriole, antidiuretic hormone, ADH, aquaporin, autonomic nervous system, biosensor, Bowman's capsule, central nervous system, CNS, collecting ducts, coordinator, cortex, cyclic AMP, CAMP, deamination, dipstick, distal convoluted tubule, DCT, effector, efferent arteriole, endocrine system, endoderm, enzyme cascade, excretion, fibrous capsule, glomerular filtrate, glomerulus, glucagon, glucose oxidase, G-protein, guard cells, homeostasis, hypothalamus, insulin, interstitial fluid, islets of Langerhans, ketones, kidney, loop of Henle, medulla, negative feedback, nephron, osmoreceptor, osmoregulation, pancreas, parasympathetic nervous system, peripheral nervous system, peroxidases, pH, pituitary gland, posterior pituitary gland, protein kinase A, proximal, convoluted tubule, PCT, receptor, renal arteries, renal pelvis, renal veins, second messenger model, selective reabsorption, somatic nervous system, stimulus, stomata, sympathetic nervous system, thermoregulation, tissue fluid, transpiration, ultrafiltration, urea, urea cycle, ureter, urine, water potential. Chapter Glossary. Abscisic acid, ABA, is a plant hormone that stimulates stomatal closing, maintains seed dormancy and triggers cold protective responses. Adenylyl cyclase is an enzyme that catalyzes the conversion of ATP to CAMP. Adrenal glands are glands which are located on the top of the kidneys and produce adrenaline and steroid hormones. Adrenaline is a hormone that is secreted by the adrenal glands under stressful conditions. It serves as a primary messenger, activating adenylyl cyclase. Afferent arteriole is the larger diameter arteriole which carries blood to the glomerulus for ultrafiltration. Antidiuretic hormone, ADH, is a hormone released from the posterior pituitary gland that increases the reabsorption of water in the kidney tubules. Aquaporin is a membrane channel used for the selective transport of water in and out of the cell. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 39. Autonomic nervous system is a branch of the nervous system that carries nerve impulses to muscles and glands. It controls involuntary activities and has two divisions, the sympathetic nervous system and the parasympathetic nervous system. Biosensor is a biological molecule is paired with a device that generates a thermal, optical or electrical signal when a specific reaction takes place in order to detect the presence of a particular chemical. Bowman's capsule is the cup-like structure at the start of a nephron that surrounds the glomerulus. The inner layer of the capsule, through which filtration of the blood takes place, is composed of podocytes. Central nervous system, CNS, is the brain and spinal cord. Collecting ducts is a system of ducts and tubules which connect nephrons to a calyx or the renal pelvis, which is a site for water reabsorption. Coordinates information from the receptors and sends instructions to the effectors. Cortex is the outer region of the kidney. Cyclic AMP, CAMP, is a second messenger involved in the action of adrenaline that activates protein kinase A. Deamination is the removal of an amino group from a molecule. Enzymes that catalyze this reaction are called deaminases. In the human body, deamination takes place primarily in the liver, however, it can also occur in the kidney. Dipstick is a device which can be used for chemical testing or to measure liquid volume. Distal convoluted tubule, DCT, is the twisted region of the nephron between the loop of Henle and the collecting duct that alters the concentration of water and salts reabsorbed. Its permeability to water is altered by ADH. Effector is an organ, tissue, or cell that produces a response to a stimulus. Efferent arteriole is the smaller diameter arteriole which carries the blood away from the glomerulus after ultrafiltration. Endocrine system is a chemical messenger system. It consists of glands, which produce and secrete hormones into the bloodstream, and effectors. Endoderm is a type of organism which can regulate its own body temperature without relying on external heat sources. Enzyme cascade, a series of successive enzymatic reactions in which the product of one reaction serves as the substrate for the next. Excretion is the process of removing waste products from an organism. Fibrous capsule is a tough layer that surrounds and protects the kidney. It consists of fibers, collagen, and elastin. Glomerular filtrate is the fluid produced by ultrafiltration of the blood into the renal capsule. It contains water, glucose, mineral ions, and urea. Glomerulus is the bundle of blood vessels at the beginning of a kidney nephron where ultrafiltration takes place. Glucagon is a hormone secreted by alpha cells of the islets of Langerhans in the pancreas. It increases blood glucose concentration. Glucose oxidase is an enzyme which oxidizes glucose to hydrogen peroxide. It is often used alongside peroxidase to determine oxygen concentration via a dipstick method. G protein is a protein that serves as a molecular switch inside cells. Activation of the G protein in the control of blood concentration results in the stimulation of adenylyl cyclase. Guard cells are cells that surround the stomata and change shape depending on water potential. They control the rate of transpiration by becoming turgid or flaccid, opening or closing the stomata. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 40. Homeostasis is the maintaining of a constant internal environment around an optimum despite external change. Hypothalamus is the region of the brain that serves as the control center for the autonomic nervous system. It is responsible for production of hormones, the regulation of the water potential of body fluids, and the control of behavioral patterns. Insulin is a hormone secreted by beta cells of the islets of Langerhans in the pancreas that decreases blood glucose concentration. Interstitial fluid is the fluid that fills the spaces between cells. It is composed of water, amino acids, sugars, fatty acids, coenzymes, hormones, neurotransmitters, salts, and cellular products. Its main function is to bathe and surround the cells of the body. Islets of Langerhans are regions of the pancreas which contain alpha and beta cells. Ketones are carbonyl compounds produced during beta oxidation of fat in the liver. A high level of ketones in the blood may indicate diabetes. Kidney is organ involved in blood filtration and urine excretion. It also produces some hormones. Loop of Henle is a large hairpin-shaped loop found in the kidney tubule used to regulate the water and salt concentration of the blood. Medulla is the inner region of the kidney consisting of renal pyramids made up of nephrons. Negative feedback is the product of a process that counteracts change to maintain an equilibrium around a normal level. Nephron is the basic functional unit of the kidney. Osmoreceptor is a type of receptor found in the hypothalamus which can detect the water concentration of blood plasma to maintain an appropriate water balance in the body. Osmoregulation is the homeostatic regulation of the osmotic pressure of body fluids. Pancreas is an organ which has both exocrine and endocrine functions and is involved in both digestion and the homeostatic control of blood glucose levels. Parasympathetic nervous system is a branch of the autonomic nervous system that is active under normal, resting conditions. It inhibits effectors, slowing down activity. Peripheral nervous system are pairs of nerves that originate from the CNS and carry nerve impulses into and out of the CNS. It is divided into the sensory nervous system and motor nervous system. Peroxidases is a group of enzymes which catalyze reactions when hydrogen peroxide is present. pH is a logarithmic scale used to measure the relative acidity-alkalinity of a substance. It is calculated using the formula pH equals log 10 H+. Pituitary gland is a small, hormone-producing gland located at the base of the brain. It is divided into two regions, the anterior pituitary and the posterior pituitary. Posterior pituitary gland is the region of the pituitary gland that stores and secretes hormones, such as ADH, produced by the hypothalamus. Protein kinase A is an enzyme involved in the control of blood glucose concentration that modifies other proteins by phosphorylating them. Its activity depends on cellular concentrations of CAMP. Proximal convoluted tubule, PCT, is the twisted portion of the nephron between the renal capsule and the loop of Henle. Its walls consist of epithelial cells that are adapted for the reabsorption of glucose and water into the blood. Receptor is a specialized structure that detects a specific type of stimulus. Renal arteries are blood vessels that carry oxygenated blood to the kidneys. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 41. Renal pelvis is the central region of the kidney that serves as a funnel for the flow of urine into the ureter. Renal veins are blood vessels that drain the kidneys. Second messenger model is the mechanism by which a hormone, e.g. adrenaline or glucagon, has an effect inside a cell by triggering the production of a second messenger such as CAMP. Selective reabsorption is the selective reuptake of useful substances, small molecules and ions, in the proximal convoluted tubule using membrane transport proteins. Somatic nervous system is a branch of the nervous system that carries impulses to the skeletal muscles. It controls voluntary activities. Stimulus is an internal or external change or factor which triggers a response. Stomata are small pores found on the epidermis of leaves that can be opened or closed by guard cells to control the amount of water loss and gas exchange. Sympathetic nervous system is a branch of the autonomic nervous system that is active under stressful conditions. It stimulates effectors, speeding up activity. Thermoregulation is the homeostatic maintenance of a stable core temperature in endotherms. Tissue fluid is a liquid formed when hydrostatic pressure at the arteriole end of a capillary bed forces fluid out into the surrounding tissue. Transpiration is the water loss from plant leaves and stems via diffusion and evaporation. The rate of transpiration is affected by light, temperature, humidity, air movement, and soil water availability. Ultrafiltration is the removal of small molecules, water, and ions from the blood in the glomerulus of the kidney at high pressure. Urea is a diamide produced from ammonia when proteins are broken down. Urea cycle is the cycle of reactions responsible for producing urea from ammonia for detoxification and excretion. Ureter is a tube that carries urine to the bladder from the kidneys. Urine is a waste liquid containing urea. It flows through the kidneys, which regulate its water content, and is stored in the bladder before excretion. Water potential is a measure of the tendency of water molecules to move from one area to another, measured in kilopascals, kPa, and given the symbol psi. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 42. 15. Control and coordination. All the activities of multicellular organisms require coordinating, some very rapidly and some more slowly. The nervous system and the endocrine system provide coordination in mammals. Coordination system also exists in plants. The differences between hormonal and nervous coordination. 15.1 Control and coordination in mammals. Nervous coordination involves the detection and response to internal changes, homeostatic regulation, and the detection and response to external changes for survival, finding food, and finding mates. A stimulus is any change which provokes a response. Receptors are specialized cells that detect a stimulus. Effectors are organs that bring about a response, muscles and glands. The nervous system can be divided into two areas: central nervous system, CNS, and brain and spinal cord peripheral nervous system, nerves linking receptors and effectors to CNS. These can be further divided into voluntary nervous system, conscious control, and perception autonomic nervous system, unconscious control, divided into sympathetic system and parasympathetic system. Transmitter chemical location in nervous system enzyme responsible for breakdown.